5:30 or 6 in the evening.
Oh,
Yeah.
that's
Yes.
all about life in US.
But it's fun, actually. I would say it's fun. It's
Okay.
fun that I have weekends with me. I have family here, so we enjoy on weekends. And California has a very good weather.
Yeah, uh,
Yes, so we go on hikes. Yeah.
yeah.
Yeah, we roam around a lot on the weekends.
So we'll start because its time is up. Okay.
Sure.
Everybody in India, is there anybody from on online or not? Are there? No, no, no one. Okay, that's good. So good morning, everybody in this hall, and good evening, Megha. So our today's speaker is Dr. Megha Agarwal. Megha, I mean, to introduce Megha, it's like she was a student in my lab, uh, and now she's a postdoctoral scholar at the Gifford Laboratory in the Department of Pediatrics, School of Medicine, Stanford University. Her schooling was in Pantnagar, BSc and H from Pantnagar, MBSc from my lab, and then PhD from the um, UNESCO Regional Center for Biotechnology in Faridabad. And the department she did her PhD was developmental genetics. She has more than 13 years of experience working with stem cells. At present, she is working on cardiac organoid and exploits the model to study congenital heart disease. She has 10 peer-reviewed publications to her credit and has reviewed many manuscripts. For every speaker, I have been curious that what prompted them to take up the current research work. I asked also the same question to her. What makes you to take up the congenital heart diseases? One reason, when somebody in the family is affected, we are more touched upon and more inspired, more motivated to do something. Megha's answer was yes. Because of the same reason, she has taken up. So, ladies and gentlemen, let's welcome Dr. Megha Agarwal for today's talk on stem cells in skeletal muscle development, regeneration, and congenital heart disease. Megha, stage is yours, mic is yours.
Thank you so much. Thank you so much for the kind introduction. Hi, everyone. I'm Megha Agarwal, as you all know, and I'll talk about stem cells in skeletal muscle development and skeletal muscle development, regeneration, and in congenital heart disease. So, the overview of my presentation will be I'll touch upon a little bit of introduction, then I'll cover all the three topics: stem cells in skeletal muscle development, regeneration, and then in congenital heart disease. And then I'll give this stage to you to ask any questions, and please feel free to ask any questions you want, either in the between or in the end, and let's make this a very productive session. So to begin with, stem cells in skeletal muscle development. So I'm very fascinated about stem cell, and my fascination started from Dr. Dhara's lab. And there, for the first time, me and you all know Saurabh sir sitting right next to Dr. Dhara, he taught me how to actually isolate porcine mesenchymal stem cells, and then we tried to differentiate them to osteoblast. So today I'll talk about more of my PhD work, what I did during my PhD, and what I'm doing right now as a postdoctoral fellow. So I'm still working on stem cells, but like Dr. Dhara mentioned, I've switched the fields. So I worked on skeletal muscle during my PhD, and now I work on heart muscle. Uh, so, so to begin my presentation, I'll just give you a brief introduction about adult stem cells. So what are adult stem cells? Adult stem cells are also known as somatic or tissue-specific stem cells. These are undifferentiated cells which are found in various tissues and organs of the body. Unlike embryonic stem cells, which are derived from embryos, adult stem cells are present throughout the life and play a crucial role in tissue homeostasis, repair, and regeneration. And upon activation, these muscle stem cells proliferate and expand to specific population, which they belong to the niche, specific niche which where they belong. So they have these unique property of self-renewal. They have this unique property of differentiation, which they differentiate to blood cells, bone cells, muscle cells, and neurons, which are depicted in this picture. And they have another amazing property of regeneration. So I've kind of studied muscle regeneration and differentiation and self-renewal, meaning all three different amazing properties of these stem cells. And I'll walk you through my entire slide telling about these stem cells and how they self-renew, how they differentiate, and how they regenerate. Now, they also have the capacity to differentiate into specialized tissues, which I'll talk about today, like in skeletal muscle and heart tissue, and they contribute, or the dysfunctional, they replace the damaged or dysfunctional cells and contribute to tissue repair and regeneration. So, like I mentioned, I'll talk about skeletal muscle. So, what is skeletal muscle? Skeletal muscles are the type of striated muscle attached to the skeleton. These are one of the largest tissue in the human body. They're responsible for voluntary movement, such as walking, running, lifting weights. Skeletal muscles are composed of muscle fibers, and these muscle fibers are arranged in fascicles. A fascicle is a bundle of muscle fiber and muscle. So here you see fascicle. These are a bundle of muscle fibers, and muscle fibers are also known as muscle cells or myofibers. And these are the individual cells which club together to form skeletal muscle. And these muscle fibers are long multinucleated cells that run throughout the length of muscle and are responsible for generating force through the contraction. So the contractile property is in the functional unit of muscle known as sarcomere. These sarcomeres are consist of myosin filaments and actin filaments, and they are formed by the Z disc. We all know this. And these sarcomeres are formed by the structural protein known as myosins. And these are formed by overlapping patterns of myosin and actin throughout the protein filament. So how is this skeletal muscle formed? The differentiation of muscle stem cells to muscle fibers is known as myogenesis. So there are these satellite cells or the stem cells. So in the adults, these are quiescent and are known as satellite cells. Otherwise, these are stem cells in the developing embryo. These differentiate, become committed to a fate known as myoblast, and these myoblasts then fuse together to form multinucleated muscle fibers. Now, how this transition of stem cells to muscle fiber takes place. There are certain unique transcription factors which are expressed at specific point during the differentiation. And here, right now, I'm talking about myogenesis. So, for example, PAX3 and PAX7 are stem cell or you can say muscle stem cell markers which are expressed during very early phase of development. And then these cells when differentiate to myoblasts express MyoD and MIF5. And then to myocytes, they express myogenin. Myogenin is the late differential marker, and myosin heavy chain are the structural proteins. And in the bottom, you see the C2C12 cell line. So this is the skeletal muscle cell line, which I've been using, or I used in my PhD. So this is how they look like. At day one, they're myoblasts, they're single cells, and then they differentiate. We kind of differentiate them. So we deprive them. We serum deprive them and give them just the hot serum, and then we allow them to differentiate, and they form myocytes at day 3 of differentiation, and then they form these long tubular multinucleated structures by day 8 or day 9, and these contract, and these muscle fiber beat in the dish actually. So this C2C12 cell line is my model system, which I use to study differentiation. Now, just coming back and going back to the in vivo myogenesis, which I just mentioned, like muscle progenitor cells differentiate to myoblasts, and then they differentiate to myofibers. And these are specific transcription factors which regulate this differentiation, and myofibers are the structural proteins which express myosin heavy chains, which are structural proteins. Now, how do you relate that to real myogenesis or during development? So during development, the myogenesis is divided into different phases like embryonic myogenesis, fetal myogenesis, and perinatal myogenesis. So here, this is the example of mice myogenesis. So embryonic myogenesis, the process begins with the specification of myogenic precursor cells, which are known as myoblasts, from mesoderm layer of developing embryo. And these precursor cells are committed to muscle lineage and then express specific transcription factors, PAX3 and PAX7. Followed by embryonic myogenesis is fetal myogenesis. So fetal myogenesis refers to the continuation and the development of muscle fiber formation during the fetal stage of development. And differentiated muscle fibers, which are known as myocytes, continue to fuse together to form multinucleated muscle fibers. And perinatal myogenesis is the dynamic process that continues to the maturation and functional capacity of skeletal muscle tissue in the newborns. And adult myogenesis refers to the process of muscle tissue regeneration, which I'll talk as second part of my presentation, which refers only to the muscle regeneration which happens in adults when there is any exercise-induced damage or pathological conditions like multiple sclerosis or in case of injury. So this is how myogenesis happens in mice. So there are different phases of development or different phases of myogenesis in mice. Please keep this in mind because I'll go back to these phases again. So like I mentioned previously, that I will talk about skeletal muscle, and skeletal muscles are defined by myosin heavy chain structural proteins. Now, myosin heavy chain, so there are different isoforms of myosin heavy chain. So myosin isoforms have different variant of myosin proteins. These isoforms are classified based on heavy chain composition and are designated as fast and slow. They're also designated, it depends on when they're expressed, like in adult, they're expressed, or if they're expressed in the developing phase. If they're expressed so there are only three myosin isoforms that are expressed during the developing phase: myosin heavy chain embryonic, perinatal, and slow. So today, basically, I'll focus on myosin heavy chain embryonic. So myosin heavy chain embryonic is a structural protein which is encoded by MYH3 gene. And this is expressed only during development. This is a very peculiar feature of this myosin heavy chain, that it is expressed only during development. And but when there was muscle regeneration, like in case of multiple sclerosis, or there was muscle injury, or there is a burn, this myosin heavy chain is expressed. Myosin heavy chain embryonic is expressed in adults as well. And here you see the example of Freeman-Sheldon and Sheldon-Hall syndrome babies which have mutations in MYH3, and you see contracture in their bones and joints. And MYH3 mutation in this gene is reported to cause this disease. But still, we do not know how this disease lead to this Freeman-Sheldon and Sheldon-Hall syndrome. So what I studied in my PhD is the role of myosin heavy chain embryonic in myogenesis. So to do this, we use different animals. Now, we generated two MYH3 animals. One is knockout, like complete knockout, and one is MYH3 embryo. One is embryonic MYH3 PAX3, MYH3 delta PAX3 mice to study embryonic myogenesis, and PAX7 MYH3 KI mice to study fetal myogenesis. Now, since most of these studies that were already done on MYH3 embryonic did not study the specific role of MYH3 specific during embryonic or fetal myogenesis, therefore, we made these targeted alleles of MYH3 to study its specific role specifically during embryonic myogenesis and fetal myogenesis. So just to explain, embryonic myogenesis. So in embryonic myogenesis, these animals will not have MYH3 only during embryonic myogenesis phase. And in fetal myogenesis, MYH3 will be absent only during fetal myogenesis, that particular time of development. So this is very important to remember. That is how we studied MYH3 roles during these different developmental phases. Now, from now on, I'll show you what we learned and what I learned after going through 7 years of my PhD. So here we quantified. So what we did is we took muscle from animals meaning the knockout animals. So here what you're looking at is the number of myofibers per unit area in the wild type animal. So wild type animal is MYH3 plus over plus. That is the wild type animal, meaning it has no abnormality, or it's like wild type. And MYH3 delta over delta is MYH3 knockout animal. And you see that EDL is extensor digitoral longus muscle, and you see there is increase in myofibers in at P0. So P0 is their date of birth. So we indicate as P1, P2, P3, or P0 is how these animals are born. So P0 is the day they are born, and we collect the embryos, and here you see EDL. So in newborns, we see increase in number of myofibers per unit area, and as well as in soleus muscle, we see increase in number of myofibers per unit area in both of these muscles in MY3 knockout animal. Now, what you're seeing here are the cross sections through the hind limb of P0 MYH3 plus over plus and delta over delta in the bottom, which are labeled by immunofluorescence for MYH3 slow in red, laminin in green, and DAPI in blue. And here you see the zoomed area of these box. Here you can see tibia fibula, and here you can see soleus and EDL muscles. And we see that, and we quantified these number of slow positive fibers per unit area, and we see increase in number of slow fibers per unit area in the knockout animal as well. And we quantified this by Western blot, and in the Western blot as well, we see there is increase in MYC slow. So MYC slow is a different myosin which is expressed during development, what I showed in my previous slide. Now, if you see that if you knock out, or in the knockout animals, there is increase in different other isoforms of MYH3. Then we looked at the stem cell markers. PAC7 is a stem cell marker. What happens to the expression of PAC7 if in the targeted knockout animal? So here what you're looking at, so PAC3 CRICAI MYH3 is MYH3 knockout only during embryonic myogenesis, which is at E13.5, and then E16.5 is fetal myogenesis, and P0 is at the time exactly when the pup is born. So you study so here we are seeing the expression of PAC7, which is downregulated in the embryonic myogenesis. It comes back during fetal myogenesis and during birth. And similarly, we looked at the expression of other transcription factors, which are myogenic regulatory factors for myogenesis. And we quantified this data here, and we see that there's initially decrease in the expression of PAC7 at early, and then PAC7 comes back. So again, to dig down, what are these cells that downregulate PAC7 or the stem cell marker very early, and then PAC7 comes back? We looked at the cross sections again for the myogenic progenitor cells. So here what you're looking are the immunofluorescence for PAC7. Now here, green is PAC7. So PAC7 is again stem cell marker. PAC7 in green and DAPI in blue, and the cross sections at E16.5 for PAC7 CRICAI MYH3 knockout animals. And these, so right two panels are the zoomed images of these sections, which are boxed in the red area. And these are the quantified images. And you see there is decrease in myogenic progenitors or PAC7 positive cells at E16.5, as well as number of MyoD positive fibers are downregulated. So then we thought that since MYC embryonic is a structural protein and it's expressed at late during the differentiation, but still it's having effect on stem cells, which are like really early in the differentiation process. So we thought to look at the C2C12 model, which I just mentioned in my previous slide. And here what you're looking are the C2C12 cells. So this is in vitro slide, meaning this we did. So these are C2C12 cells, and we did this culture in a lab. And we then looked at, and then we looked at the stem cells. So these here, all the arrows indicate stem cells. So the round cells, when we culture C2C12 cells, are all stem cells. And so here what you see are the Western blots for MYC embryonic, MYC slow, and beta actin. when we treated these cells with control siRNA and MYS3 siRNA. So this is the in vitro slide, like I mentioned. And in the previous slide, I showed that in the knockout animals, there's decrease in PAX7 positive cells. Now we try to recapitulate this in vitro system. So in vitro system, we used siRNA to downregulate MYS3. And here, what you're seeing are the Western blots at day 1, 3, 5, 7, and 9 of differentiation. And C stands for control siRNA and S stands for MYS3 siRNA. And days are depicted here on the top of the Western blots. And this is the densitometric quantification of these plots. And here it clearly indicates that there's decrease in MYC embryonic and there's increase in MYC slow, which I showed in my first slide. So this correlates with the in vitro and with the in vivo data that upon downregulating MYC3 or MYC embryonic, we see increase in MYC slow. And in the left panel, you see the immunofluorescence images, which are stained with phalloidin. So phalloidin stains actin fibers, and the round phalloidin at very early stage of differentiation are stem cells. And so the green stains phalloidin and blue stains TAPI in the control and as well as in the MYS3 siRNA. And this, we counted these number of reserve cells or stem cells, and we see there is decrease in stem cells if you downregulate MYS3. Now, again, so till here, we were sure that MYS3 is doing something to these stem cells, and MYS3 is the structural protein which is expressed in the differentiated fibers. But how is it controlling stem cells, which are like really very first or are very first in the differentiation process? So then we thought that there is something going from these muscle fibers to these stem cells, which makes these stem cells either it's killing these stem cells or either it's reducing the proliferation rate of these stem cells. So what we did is we did a very innovative experiment. So we treated these cells with siRNA, then we collected the media from the cells treated with siRNA, and then we treated these cells with that media which had MYS3 siRNA or control siRNA, and this is known as conditioned media. So here you see immunofluorescence images labeled with MYC embryonic and DAPI in blue on C2C12 cells which were differentiated for 4 days in the conditioned media from control siRNA and MYS3 siRNA, and we quantified the fusion index. So fusion index is number of nuclei per fiber as a fraction of total number of nuclei. And here you see that if in MYS3 siRNA treated media, we see increased number of myofibers when we compare them to control myofibers. So it means that there is some non-cell autonomous signal mediated from MYC embryonic that is crucial for differentiation, and that is why we see this difference in fusion index. And then we quantified this data, and we looked at different signaling molecules or different pathways which we thought that might be important or might be regulating this non-cell autonomous function. And we see that there is this FGF signaling proteins or FGF signaling pathways which are downregulated in the knockout animals, and this might be involved in the non-cell autonomous function of MYC embryonic. So what we also did is we did this heat map. So here what you're seeing is the mass spectrometric. So this is the heat map from the mass spectrometric analysis of secretome. So like I said, what we did is we treated C2C12 cells with control siRNA and MYS3 siRNA, and then we collected the secretome or the media, and then we ran mass spec with that secretome, and we showed here the abundance of FGF ligands in the control siRNA when you compare that to MYS3 siRNA treated C2C12 cells. So again, this was very assuring that there is this FGF signaling which is regulating the non-cell autonomous function of MYC embryonic during development. Then what we did is we tried to see that whether if we exogenously give FGF, can we rescue this phenotype? So what we did is we supplemented these cells with FGF, endogenous FGF. So we can get FGF from locally available or commercially available vendors, and then we got this FGF. We treated these MY3 siRNA cells with and without FGF. So the top panel is without FGF, and the bottom panel is with FGF. And then we again quantified these phalloid. We immunolabeled these cells with phalloidin, and then we quantified stem cells. So these round cells that you see are all stem cells. And then we quantified fusion index, which is again the marker of differentiation, because fusion index tells you how well these cells have differentiated. And we see that upon treating with FGF, the fusion index, number of reserve cells has increased, and the fusion index is being restored. So this made us conclude that FGF signaling is the one which is regulating the non-cell autonomous function of MYC embryonic from muscle fibers to stem cells. And here is the model which we proposed. So this is the model summarizing the non-cell autonomous and cell autonomous uh, functions of MYC embryonic. Here we propose that MYC embryonic is very crucial for cell autonomous and non-cell autonomous functions during embryonic, fetal, and neonatal myogenesis. MYC embryonic within the muscle fibers during embryonic and fetal stages is required to regulate FGF levels, which control the rate of differentiation of muscle progenitors and myoblast into muscle fibers. Reduction of FGF. So here blue dots are, or blue stars are FGF. So reduction in FGF levels cause accelerated differentiation of progenitors to the myoblast and then to the myofibers, which depletes the pool of progenitors and myoblast. So this is the non-cell autonomous function of MYC embryonic. So I don't know if I mentioned, so cell autonomous function meaning the effect of that gene or that protein on the same cell type, but non-cell autonomous function is the effect of the gene or protein on a different cell type. So this is what we explain in this paper, saying that MYC embryonic has dual role of cell autonomous function, where it controls muscle fibers, as well as non-cell autonomous function, where it controls stem cells or the progenitor cells. And we also show that MYC embryonic regulates the levels of other myosins, as well as fiber types, number, and size, which I showed in my first slide, which is all the autonomous roles of myosin heavy chain during embryonic, fetal, and neonatal myogenesis, where it regulates the FGF levels, which in control regulate myogenic differentiation. And we published this paper, I think now 4 years back. So this was my first part of my presentation, where I talked about myosin heavy chain embryonic and its role during muscle development. So now the second part of my talk is stem cells in skeletal muscle regeneration. So skeletal muscle, yeah,
Can I stop you for a moment?
sure.
When your data shows that FGF receptors are absent in the knockout animal,
Mm-hmm.
then supplementation of FGF, how does it risk?
Sorry?
So in this data, it shows that in this figure,
Yeah,
that FGF receptors are absent.
yes.
Then supplementation in the next slide, supplementation of FGF is rescuing the some of the cells. What you say,
Yes.
stem cells. How does it work?
So this is what we show, that FGF is controlling. So FGF controls stem cell population, PAX7 positive population. So because this FGF is coming from fibers, and fibers express myosin heavy chain embryonic. Now, if there is no embryonic, there is no FGF. And when there is this recruitment of FGF, when it's altered, then it has effect on progenitors or stem cells, because FGF directly controls the levels of stem cells or PAX7 positive progenitors, muscle progenitors.
Okay, so the maybe questions maybe okay. We'll save it for the next time. Okay, no issue.
Okay.
Thank
Okay. Okay. So Okay, now coming to my second part of the presentation, where I'll talk about stem cells in skeletal muscle regeneration. So skeletal muscle regeneration is seen in adult muscle fibers and in the adults. So this is skeletal muscle satellite cells or stem cells are major contributors for muscle regeneration, and muscle regeneration is seen in during accident, during severe burns, or in multiple sclerosis. So what you're seeing here are the muscle fibers here that are labeled with PAC7 in red, and these muscle fibers are PAC7 positive fibers, and these PAC7 positive satellite cells are really important for muscle regeneration. So this is the example of how important these PAC7 positive muscle progenitors or stem cells are. So in this animal where you see the ablated PAC7 positive satellite cells, you see the muscle did not. So here you see this muscle has completely regenerated upon injury. But in right panel, you see that the muscle did not regenerated in PAC7 ablated animals. So this clearly indicates that PAC7 positive satellite cells are very important for muscle regeneration. So these satellite cells undergo self-renewal or commitment upon activation. These are the satellite cells which are present in the adult muscle, and they are sitting there quiescent, which express PAC7. But upon activation, like during injury, during exercise, or in maybe severe burn, these satellite cells get activated, and these activated satellite cells express PAC7, MIF5, and MyoD. These satellite cells then either self-renew or go back and form quiescent satellite cells which express PAC7, or they become committed to form myocytes and undergo regeneration and help in myogenesis. So, and these myocytes express MIF5 and MyoD. Now, what I want to study was that how this decision, these stem cells take the decision of whether they want to differentiate or whether they want to go back to the quiescent state. And so during my PhD, I also studied a family of proteins known as transducing like enhancer of split family of proteins, and these are co-repressors. So I'll tell you in my subsequent slides what these proteins are. So other than transcription factors, there are co-regulators that play very important role during transcriptional regulation. So yeah, here yellow is the co-transcription factor and green is the co-regulator. So co-regulators are the proteins that do not bind to the DNA directly, but bind to the transcription factors and hence control transcription regulation. And Gaucho is one of the well-known developmental co-repressor. So Gaucho or transducing like enhancer of split TLE family of co-repressors are the evolutionary conserved developmental co-repressors. These play a very important role in Wnt and Notch signaling. They are important for segmentation and neurogenesis in Drosophila. And in vertebrates, they are known as transducing like enhancer of split. And there are 4 full-length transducing like enhancer of split isoforms present in humans. So this is how the structural or the kinetics of the TLE protein looks like. And there was this, like all the proteins, they have this N-terminal TLE domain, the unstructured domain, and the WD40 domain. And WD40 domains are the part of TLE proteins that interact with the transcription factors and which in turn bind to DNA. So TLE proteins are known to play several roles during development, and like they are known to play role in pancreatic development, liver development, kidney development, and thus so individual TLEs are expressed in distinct patterns during the development and they perform specific function. They are also known to play important role in several diseases like cancer, in neurogenesis, adipogenesis, osteogenesis. So this has already been known. So what was not known when I was doing my PhD is the role of TLE in myogenesis. So to begin with, we started to look at the expression of TLE4. So today I will talk only about TLE4 as I completed this project, but during my PhD, I studied all the TLEs. So I will today specifically talk about TLE4. So to begin with, we looked at the expression of TLE4 in C2C12 cells, the same model which we used to study myogenesis. And here you're looking at the expression of TLE4 at day 1 and day 5 of differentiation, stained in red, and the phalloidin which stains actin fiber in green and DAPI in blue. And the right side are the zoomed panels, and here you see that TLE is localized in the nucleus. So here and in the bottom panel, you see qPCR, where it shows the expression of TLE4 throughout the course of differentiation and at the protein level as well as at the RNA level. Now, since I'm interested in studying muscle regeneration, so we wanted to look at how to study this muscle regeneration by using mouse as my model. So what we do is we inject barium chloride to the mice TA muscle, and then we isolate this TA muscle from the mice at different time points. So 3, 5, 7, 14, and 28. And so DPI stands for days post-injury. So this is how we study muscle regeneration using mice as our model, and we inject barium chloride in the left thigh, and right thigh we use as the control. And these are mostly all, so whatever data I will show you are from TA muscles of mice. So here what you're looking at is, so U stands for uninjured muscle, I stands for injured muscle, and this is the Western blot. So this is the Western blot showing the expression of TLE4 throughout muscle regeneration at day 3, 5, 7, 14, and 28 day post-injury. Here you're looking at TLE4 and PAC7 expression. So PAC7, again, to remind you, is a marker of satellite cells or the muscle stem cells. And we quantified this data, and we see more or less the expression pattern of TLE4 and PAC7 coincide with one another. Like initially there was increase in the expression of TLE4 and PAC7. And when the muscle is regenerated at around day 14 and 28, we see there is decrease in the expression of TLE4 and PAC7 both. And we thought that these two proteins are really expressed very high levels just after the injury. They're there during muscle regeneration, and the expression goes down immediately when the muscle is regenerated. So, and since TLE4 is a co-repressor and PAC7 is a transcription factor, so we hypothesized that these might interact. So then we did immunoprecipitation assay. And so this is the co-immunoprecipitation of MYC-PAC7 and FLAC TLE4, which are expressed in NIH 3T3 cells. And here also you see PAC7 and TLE4 in undifferentiated C2C12 cells. And you see that these both interact. So this gave us a proof that TLE4 might be involved in muscle regeneration, or it might play some role during muscle regeneration because it interacts with PAC7. And the expression pattern of TLE4 also looks like more or less similar to that of PAC7. So then we looked at the expression of TLE4 during muscle regeneration. So here what you're looking at are the cross-section of TA muscles. So the left panel is uninjured. The top panel is uninjured. The bottom panel is 5 days post-injury injured and 14 days post-injury injured. And these are labeled with TLE4 in red, PAC7 in green, and then this is DAPI, and the last panel is merged image. And in the boxed, you see the zoomed images of these squares. And here we see that upon muscle injury, TLE4 is transiently downregulated in subset of satellite cells. Here you see these two satellite cells, which are uninjured. And here these two satellite cells, this satellite cell express TLE4, but this satellite cells do not express TLE4. And then at day 14 DPI, you see these both cells regain TLE4 expression. So there are some cells that upon injury or during regeneration downregulate TLE4 transiently. So what are these cells that are downregulating TLE4 transiently? And we quantified this data, and we see that, so in the red bar, you are seeing number of PAC7 positive TLE4 negative cells. And you see that TLE4 negative cells are more during initial time points, and then they come back to that of uninjured levels. So we thought that what are these cells that are downregulating TLE4 transiently? And then we labeled these muscle fibers with MyoD. And so here what you're seeing are the muscle fibers, which we labeled with TLE4 in red, MyoD in green, and this is the merged image. And we quantified this data, and we see that at 12 hours or 24 hours post-injury, there are very less or very low number of TLE4 positive, MyoD positive fibers. So this kind of indicated that TLE4 is transiently downregulated in activated satellite cells. So now satellite cells are also of two types. There are activated satellite cells which will undergo differentiation, and there are satellite cells which will undergo self-renewal, like I showed in my previous slides. So since MyoD is a marker for differentiation, and we see that TLE4 is downregulated in MyoD positive cells, so this led us to conclude that TLE4 is transiently downregulated in activated satellite cells. So now to prove this, again, we use C2C12 as our model, in vitro model, to just validate our hypothesis. So here what you're seeing are C2C12 cells at day 5 and 7 of differentiation, and these are labeled with uh, myosin heavy chain in red and actin fibers in green, and we quantified the fusion index. So like I mentioned in my previous slides, fusion index is the number of nuclei in one fiber as a fraction of total number of nuclei. And we see that the fusion index is dramatically downregulated in TLE4 siRNA treated cells when you compared with cells with control siRNA. So it means that TLE4 is required for proper muscle differentiation in vitro. So then we looked at myogenic regulatory factors like MyoD, MIF5, and myogenin. And here you see that most of these transcripts, so these are the Western blots, and these graphs are the densitometry for these Western blots. And here we see that TLE4 is downregulated by siRNA, which is ideal, which should be the case. And then we see there is increase in MIF5, increase in MyoD, and increase in myogenin. So MyoD and myogenin are all so myogenin is basically more terminally differentiated marker, and MIF5 and MyoD are intermittent expressing markers for myogenesis, and myosin heavy chain embryonic and slow are structural proteins, like I already mentioned in my previous slides. So most dramatic effect was on MIF5. There was increase in the expression of MIF5 upon TLE4 downregulation. Then we thought that since TLE4 is a co-repressor and MIF5 is a transcription factor, so TLE4 might be somehow regulating expression of MIF5, and we already know that TLE4 interacts with PAC7. This I showed in my previous slide. So what was already known is that PAC7 has a binding site on MIF5. So this is the schematic representation of MIF5 enhancer, that is 111 kb upstream of transcription start site, where PAC7 binds and activates MIF5. So now TLE4 interacts with PAC7, which we have already shown, and PAC7 activates MIF5. So what's the relationship between, and TLE4 regulates MIF5 by my qPCR. So then to prove this, we did chromatin immunoprecipitation assay or CHIP assay. And here we see, so green is control and red is TLE4, and we see TLE4 co-occupies this minus 111 kb enhancer region along with PAC7. So this proved that TLE4 represses PAC7, which is mediated by MIF5. We also did a luciferase assay. So this is the quantification of the luciferase assay at minus 111 kb MIF5 enhancer. So if you look at this blue bar, so in presence of TLE4, you see decrease in the activation of MIF5 and MIF5 enhancer. But if you look at this red bar, in the presence of TLE4 and PAC7, sorry, in the presence of TLE4 and PAC7 both, there is decrease. I can go back, yes. So in the presence of TLE4 and PAC7 both, there is decrease in the activity of MIF5 enhancer. So this made us to conclude that TLE4 represses PAC7, which is mediated by MIF5. Then we wanted to test the role of TLE4 in muscle regeneration in mice. So what we did is we used control siRNA and TLE4 siRNA, and we injected siRNA in the TA muscle of mice. And then we looked at the expression of these myogenic regulatory factors and myosin heavy chain embryonic. And we see that here is the quantification, and we see that there is decrease in the expression of myosin heavy chain embryonic. We then also looked at the number of satellite cells upon TL4 knockdown given siRNA injection to the animals. And here, what you're looking at in the top panel is the control siRNA injected TA muscle of mice. The bottom panel is TL4 siRNA injected TA muscle of mice, which were labeled with PAC7 in red, and laminin, which labels the sarcolemma of muscle fiber in green. And you see that this we quantified here, and you see that there is significant increase in the number of satellite cells in siRNA in the muscles which were given siRNA, TL4 siRNA. Then we also wanted to see that what's happening exactly to the muscle regeneration if you do not have TLE4. So in the top panel, you see control siRNA treated cells, and in the bottom panel, you see TLE4 siRNA treated the muscles which were given TLE4 siRNA in the animals. And so serious stress is the marker for muscle fibrosis. And you see there is increase in the muscle fibrosis if you inject these muscles with TL4 siRNA. And we quantified this data here. So this indicates that loss of TL4 function lead to altered muscle regeneration, because you see there's increase in fibrosis in the TL4 siRNA injected cells. And hence we propose this model summarizing this part of the study by saying that in quiescent cells, TL4 occupies this minus 111 kp PAX5 enhancers along with PAX7 and inhibits the activation or immature activation of satellite cells. But when there is activation of satellite cells, there is downregulation of TLE4, which leads to PAX7 binding to the MIF5 enhancer and hence activates transcription. So this is what we concluded this part of my work, and we published 2 years back. So this concludes the second part of my talk, where we show that TL4 regulates muscle regeneration via PAC7 and MIF5 enhancer. So now coming to my last part of my talk, where I'll talk briefly about stem cells in congenital heart disease, which I'm studying right now. So just to give you brief upon what are congenital heart disease. So congenital heart disease are any heart disease which when a baby is born, it has a defect in the heart, is congenital heart disease. And this is most common congenital malformation, which is present in around 1% of live birth. And this is the umbrella term for many different for many different structural defects present in different parts of heart. This is the leading non-infectious cause of death in first year of life. This is genetic because this is very common in first degree relatives. Now, unfortunately, while we know that this is very common, it is very difficult to identify the definitive cause of this disease. So now on the right side, what you're looking at is the pie chart, which indicate that causes of congenital heart disease. So here in dark red, you see there are only 5% known cases which have single gene mutation effect that cause to congenital heart disease. So all other these defects like copy number variants in 22q11 or trisomy 13 or 18, these are all associated with congenital heart disease, but still we do not know the definitive cause of around 95% of congenital heart disease. Therefore, it's very important to know the causes of such common or the most common congenital malformation. Now, to know the cause, we should know how heart is formed. So this is the summary of a mouse heart formation. So this is the cardiac crescent at E7.5 in mice. These cardiac crescent then undergo morphogenesis, form a linear heart tube, which undergo segmentation to form multi-chambered heart formation at around E10 to 12.5, and these mature to form septas and all four chambers of the heart by birth. Now, these formation of valves, ventricles, atria, and conduction system are under the control of regulatory proteins that act independently or that act in via a common pathway. For example, NKX2, 5, MESP1, myocardin are all present and are very important during cardiac crescent, and then GATA4, MESP1, and these are all the transcription factors which are very well known, very well studied, and are important during different phases of heart development. So today I'll touch upon briefly upon one of the transcription factors known as NKX25, and mutation in all these transcription factors are associated with congenital heart disease. Now, congenital heart disease affects most part of the heart and can be classified into three broad categories: cyanotic heart disease, left-sided obstruction, and septation defects. And NKX25 mutations are all associated with diverse lesions, or for example, teratology of Fallot, HLHS, atrial septal defects, etc. So this indicates that mutation in only one gene is responsible for such a varied phenotype, in which is the umbrella term for congenital heart disease. Now, why it is so difficult to study this disease, and why we, after so much of development, why still we do not know the exact cause of this disease? Because we still do not know what's the right model to study congenital heart disease, and we still do not know which cell types do congenital heart disease arise. So here I show you the heart and the heart wall. So this heart wall is formed of endocardium, which is the inner lining of the heart wall, then the myocardium, and the outermost is the epicardium. And this heart wall is composed of several cells, several different types of cells like smooth muscle endothelial, fibroblast, and cardiomyocytes. So there are so many cell types in the heart walls. We do not know what exact cell type do we study and from what cell types does CHD arise. And here in the right panel, I show you the different endocardial phenotype. Like this is the example of wild type heart, which is compared to HLHS heart. So HLHS is left-sided hypoplastic heart. And here I'm showing you the endocardium, which is completely gone, or there is very less endocardium, which you see very nicely formed here in the wild type heart. It's not there in the HLHS heart. And here you see that cardiomyocyte as well as, so here in the greens, so green signal indicates cardiomyocytes. And here you see that cardiomyocyte as well as endocardium both are affected in LVNC heart. So LVNC is another cardiomyopathy, which is known as left ventricular non-compaction, and it impacts and it so the heart muscle remains immature and cannot pump blood normally. So here also we do not know what are the cell types that are involved, but we know that cardiomyocytes and endocardial and endocardium of the cell of the heart walls are affected. So now to study this, what we have come up is we make three-dimensional organoids. So these are, we call them cardioids because these are three-dimensional organoid. And what we do is we aggregate these cells in the 9612 plate, and then these form self-aggregating or organoid bodies. And then we allow them to differentiate by regulating different signaling pathways like Wnt signaling or TGF-beta signaling. And this is how you see a beating cardioid. So we see them beating in the dish. So this by cardiomyocytes, because contractile beating is the hallmark of cardiac function. And this is how we study cardiac differentiation or congenital heart disease in tubes by using induced pluripotent stem cells. So why I say induced pluripotent stem cell? Because this is the family. So this is the family which I study right now. So the family members, so the parents. So what you're seeing here in the blue is the genotype of the pair of mother, and this is the genotype of father. And here you see they have three children, and all the three children have LVNC, which is left ventricular non-compaction. But parents are completely fine. Parents are normal. So father has only one mutation in NKX25, and mother has two mutations in MKL2 and MYS7. But the children's and they both are normal, but they had three children, and so one passed away. So they had a boy that passed away, but two girls they have, and both the girls were born with left ventricular non-compaction. So what we did is we isolated uh, skin cells from these, I should not call them, they are doing fine now, from these individuals or these girls, and then we made iPSCs from those heart cells. So these are known as induced pluripotent stem cells. And now we use these iPSCs or induced pluripotent stem cells to study congenital heart disease. So, and just to tell you how the left ventricular non-compaction muscle looks like. So this is the normal muscle in left ventricle. But what happens is that uh, during the normal developmental progression, the trabeculation becomes compacted, transforming this heart muscle from sponge-like to smooth and solid. But in LVNC, this doesn't occur. There's no compaction, and that is why you see these trabeculation or hypertrabeculation in the LVNC patient, and that is why it's very difficult for them to provide enough oxygen while they exercise or while they do heavy work. So uh, it's very important to study these disease like we went through this family, and the parents were normal, and all three children had this disease. So what I do is I make cardioids or heart organoids, and this is how they look like. So I make these organoids, I do a time series study, I isolate these cells at different time points and look at different markers of different cell types. So why do we study these three-dimensional organoid? Because in these three-dimensional organoid, we can study different cell types like endocardial cells, cardiomyocytes, fibroblasts, smooth muscle, and this cell-cell crosstalk is very well defined and very well studied in these three-dimensional organoid. So this is why I switched from two-dimensional to three-dimensional differentiation. I'll show you just one or two slides of my preliminary data, what I see when I differentiated these cells or made these organoids out of these iPSC cells. So in top panel, you see unaffected cardioid or these organoid from unaffected individual or the mother. So we take iPSCs from mother, differentiate them, and then we compare these iPSC, differentiated iPSC, to the patient which had childhood onset of LVNC, and we label these organoids or these or sections of organoids with ERG. So ERG is the endocardial marker in red, and GATA4 is endocardial as well as cardiomyocyte marker in green. And we compare this in unaffected as well as childhood onset LVNC, and we see that there's decrease in the endocardial cell population in LVNC lines. So this is at day 2.5 of differentiation. And similarly, I see at day 8.5 as well, there's decrease in the endocardial cell number. So this is right now ongoing study and ongoing work which I'm doing right now, and maybe soon we'll come to a conclusion or we'll come to a very novel finding of a better way to study left ventricular non-compaction and congenital heart disease using stem cells and organoid as my model system. So this is my last slide where I would like to conclude. And before concluding to the final, or before showing the final slide, I would like to acknowledge my PhD mentor, Dr. Sam, and his lab, and the funding Wellcome Trust, DBT Alliance, DBT DST, RCB, and ICMR. I was ICMR fellow there, and right now I would like to thank Dr. Casey Gifford. She's my mentor right now, and all the lab members, and the funding from Stanford University BASE Initiative. So BASE Initiative is a basic science and engineering initiative where they give like initial startup funds to the labs, and my lab is part of that. And I have American Heart Association postdoc fellow, and I'm supported by AHA grant. So this is all, and I would like to thank everyone who were like part of this work which I just presented. And I would like to thank the audience, and I'm open to any questions if time permits. Yes, we have 3 minutes or maybe more.
Thank you, Megha. It was a sort of as if we are watching the movies actually going on. Wonderful, very nice, very nice. Huh?
Thank you.
So, if anybody has got any questions among in the audience. Okay, if nobody has got a question, maybe I had some few things to ask about.
Yeah.
Megha, about did you have ever considered? I'm talking about the last part of your work, like
Okay.
congenital heart disease. Did you consider an experimental model to include the parents, like organoids, how they because the single.
Yes.
Go ahead.
Yes. So the slide which I showed had unaffected is the parent.
Okay, so the
Yes. So
carry two different mutations, so basically three different mutations. One with a single
yes.
mutation, and the other is the with the two different mutations.
So father had one mutation, mother had two mutation, and they both are normal. And so father had mutation in gene A, mother had mutation in gene B and gene C. and babies had mutation in gene A, gene B, gene C. So babies all uh, took the mutation from mother as well as father. Now we compare mutation from mother to the daughter and mutation from father to the daughter. So it's like, yes, so we compare like both mother to the daughter and father to the daughter.
Okay, so the question next comes: are these genes located on the X chromosomes?
No.
And then why the boy was dead?
Yes. Yes, we don't know.
We don't know. So
Yeah.
it's so so are you going to collect now more samples with an individual mutations?
Yes. So individual individual mother we can have because she has only one mutation. Now father we cannot, but I'm making those mutations in vivo in lab.
Okay, that's good. Wonderful to know, Megha, and good to see you. Actually, this is
Good to see you as well.
yeah, wonderful. Again,
Thank you so much.
it's already 10:30 in your what's time now? On
It's 9 p.m.
30, okay.
Not that late.
Okay, not that bad. Okay, good.
Yes.
We'll be in touch
Yes.
and we'll close it for the time being and we'll be in
Sure.
touch. Yeah.
It was nice.